So we come to our next speaker, Hans-Jörg Scherberger. Actually, he shares a common feature with Nikos in a way, maybe many, I do not know, but at least one I saw. He did a Master of Mathematics in 93, and then he did a medical doctor in 96 at the University of Freiburg. So, master in mathematics. So, I also have one, by the way. So, he is now head of neurobiology of the neurobiology lab at the German Primate Center in Göttingen, and he is also professor of neurobiology at the University of Göttingen. So, please, you're ready?
I think I am, hopefully, yes. Thank you very much for the nice introduction. Thank you for the invitation to talk today, and thank you for Nikos. I think I can't avoid that if I get this to work. Should work in a moment. Now, because I met you, I think, a long time ago when I was still studying mathematics and medicine, when Ernst Pöppel organized a meeting in Jülich in 1990 or 1991. And I didn't realize it at the time, but when I later looked at the book, you had an article in there. So I have met you then, and we interacted very much. But I never worked with you, so I kept the distance, but I followed quite a bit your impact to the field, and you were quite an inspiration for young people, I have to say. So why? Because he tackled really the hard problems with your background, you know, from mathematics, from physics, and then you tried new things, developed new methods. That's what's progressing science. You produced great science, excellent science. And what struck me most over the years is you really kept your curiosity, right? You could be really intrigued by some detail, some question like we saw right now, thinking across different directions, and I think that characterizes you, and that makes you an inspiration to many of us. Thank you very much for having us. The topic today is the mystery of the brain. And the first question we should ask is, why do we have one? We got some hints in this meeting. One is that we might have a brain, or we should have a brain, because we move. So we move, that is the distinction from animals to plants. And we move as humans, we move in a very sophisticated way. And so the entire capacity of the brain in primates and other animals too would be to use hints and information from the environment, now and in the past, to be prepared to do the right movements when it comes to that. And that's why we have memory, that's why we have emotions, that's why we have maybe pain and other things that guide us in doing the movements. And one important movement that we are doing as humans and as primates are hand movements, and that's the topic of my field, of my studies. And you all do that all the time. You know more about hand movements as you probably would admit. You have the capacity to, do maybe sophisticated things like handwriting or working with a hammer, or if you are educated well enough for that, you can play the piano. And you have quite a development of this instrument, the hand, throughout your life, right? You come with a small device, and when you grow up and become an adult, you have a big device. And you have to change your brain to deal with it, right? It's something that is not stable as movements, and the body is not stable at all anyway. So your brain has to adapt to this device, to the hands, throughout your life. And of course, you learn how to interact with the world, how to use the fingers, the hand. You do that in the first year and the first years in the childhood. If you touch things, you learn the things, but you also learn how to interact with them. And that's important. And it is important sometimes for the survival of a being to do the right things, to push the emergency button or to get out of something. And sometimes it's even important for a nation, whether or not that thing didn't work, huh? Whether or not whether or not a certain hand movement is going in the right direction or not. And so the question we should pose is, what is the sensory motor transformation? What is the actions that the brain does to generate hand movements? So, and that's basically what I'm studying. What brain parts are involved in performing hand movements? How do we use sensory information like vision, but also touch, from the environment to interpret, to make hand movements work? And how is that then sent down to the motor centers to move the muscles and to make meaningful hand movements? And at the cortical level, at the higher level of how this sensory motor transformation works, we have to deal with quite a number of brain areas. And I'm not saying that we are really knowing all of them and we know the entire machinery, but at least 3 brain areas are involved, are important. One is in the parietal cortex, the interparietal area, which, as we will see, is important for the selection of shape and orientation of objects to be interacted with. Then there is the premotor cortex, in particular the ventral premotor cortex, that is important for the selection of grip types required to grasp the object. And then there's motor cortex, and in particular the hand area, that is important for executing the movement. And that sends down then signals to the spinal cord. And of course, I'm ignoring a lot of activity in the spinal cord that deals with the direct activity of motor neurons in the spinal cord. What I will talk today about is the cortical control of grasping, and one part is about the coding properties of these neurons in these certain parietal and premotor areas, how these grasping movements are encoded. And then we can ask, and that's the applied part, to ask how can we make use of these signals, how can we decode them to control neuroprosthetic devices. And of course, this has to start with the understanding of the physiology, and then we can deal with the problems in implementing this for patients. And as a third part, I think because we see this development of going from the study of single neurons to populations of neurons, we also should look at the neural network properties of this network, as we can do as an example, but this can be done, of course, with other networks as well of the brain. How do neural populations in the brain functionally connect to each other to achieve functions? So if you want to study grasping in primates, we are really bound to do that in monkeys because that's the thing, the appropriate animal to look at. Nowadays, there are more and more studies also for looking at grasping in rats and mice, but the versatility of these grasping movements are rather limited, at least in my ignorance. To study this in primates, we developed the data glove to look at the kinematics of the fingers and the hand. And of course, we put them in a primate chair and put them in front of a manipulandum and train them to grasp these objects. And once we have done that and do that in a delayed grasping task, we can then train them to do and indicate different grip types, like here a precision grip, like grasping a raisin, or a power grip, like grasping a branch. And here's a neuron from, I think, parietal cortex, AIP, where we see spike rasters as blue rows of dots, and then peristimulus time histogram as a firing rate over time. And you see here this neuron, it's the same neuron on the top and the bottom, is basically silent during the baseline period, becomes strongly active when an indication is given, a cue of what grip type to make, and then the activity stays up until the movement is made. And the same neuron is only very briefly active and much less active when, instead of a precision grip, a power grip is made. So we have a clear selectivity or tuning for this particular grip type, for this particular neuron. And you can then record for many neurons and make a paper out of it, or you can also look at different features. Here, for example, is a variation of the orientation of this handle that is grasped from upright, vertical, to turning to the left or turning to the right. And you can see here, this neuron has a preference for precision grip versus power grip, but it also has a strong preference for the orientation of the object, because it is much more strongly active in a vertical orientation and not at all for a tilted orientation to either side. And then you can write a paper about it. But you can also go further on and ask, as we did, for example, what is the variation of the location in space in these grasping areas? So these areas have been described by Sakata and Rizzolatti on being specific for grasping, as I just showed you. But if you change the location of the target from straight ahead to the left and right and up and down, you see a variation of the response of this same neuron to the same grip type, to its preferred grip type, from being very strong to very weak, at least during the preparatory phase. During the movement phase, you see the activity getting up. And there's also similarly a variation on the gaze position, so you keep the target at the center, but the monkey is forced to look to the left and to the right and up and down, so we see a variation of gaze position in the same neuron. And to be sophisticated, it might be a retinotopic position of the target location in space that is seen here to be brought together. But we have a clear variation or clear tuning and selectivity of this neuron, not only for the grip type, which I don't show here, but for the location in space. And you can move on. You can see here a study where we changed the handle from a solid handle to a rubber tube from a bicycle tube that was inflated so we can measure pressure. And then you can force the monkey or ask the monkey to make light movements or more forced, stronger movements. So the pressure of the grip has to be either little, medium, or high. And then we see that these neurons in premotor cortex and also in parietal cortex, they also encode the intended force of the grip, which you might think is not so far away, but it's encoded there. And we can move on and study the laterality of the hand. You might think that it's a strong contralateral representation. That's true in motor cortex. But in premotor cortex and in parietal cortex, we also find a strong ipsilateral representation of the grip, so ipsilateral orientation of the grip. So here we don't see much difference here in A5 for contralateral and ipsilateral movement, and the same is true for AIP, and the same is true also for orientation of the object. So that means there is also a coding or a representation of ipsilateral movements there. And then we can also move on and study the amount of reward that is to be expected from doing a movement. So normally you do something for a purpose, and then if you know already whether you get a large reward or whether you get a small reward, changes your, as you can see here, makes a strong difference in the activity of the neuron in the preparatory phase. So here is the cue phase, the memory or planning phase, and here's movement execution. And we see here these neurons in F5 as well as in AIP, for example, are modulated by the expected reward. Often is up higher activity, but sometimes also small reward has a higher activity. So we see a representation of expected reward in these neurons. And you are probably all familiar with the concept of mirror neurons, discovered in Parma by Giacomo Rizzolatti, but also been studied here in Tübingen some time ago, where neurons are not only active when a grasp movement is happening, but also when the animal is observing either a conspecific or a human experimenter grasping something, but the monkey is only observing the movement, and still these neurons fire. So observed actions are encoded in these brain areas. Together, we see here that we have a simultaneous representation in many times in the same neuron, but often at least in the same area, of grip type, object properties like orientation, reach location and gaze position in space, grasp force, hand laterality, expected reward, and observation of others' actions. So we can summarize that as that these cortical areas often represent a multitude of signals in parallel. We have a mixed representation or mixed coding. How can you ever expect to decode something meaningful out of that, right? But I just showed you examples of individual neurons that have their idiosyncratic or their individual combination of selectivity for some of these properties. And the brain, of course, can deal with it by taking a population into account and then you can kind of subsumize or integrate across these different individualities. The other question, of course, is if you think about these brain areas representing object properties versus representing movement plans, is what is really represented. If you just think about grasping a simple object, that could be the activity you see could be a representation of an object property, or could already be a motor plan that is prepared there for being executed, and we should be able to distinguish that. In order to do that, we need to look at many objects, not just one with two grip types, but many objects. We definitely need to monitor the movements that are happening, so the nuances of the hand movements, the finger movements. So we need to have a means to measure the kinematics. And as I mentioned, we need to study many neurons at the same time. We need to look at populations of neurons. And for that, we need ways to record for many neurons at the same time, simultaneously. So Stefan Schaffelhofer from my lab at the time started this project where he, first of all, designed about 50 objects of different shape and size and thickness and orientation that you see here. So here are, for example, some small objects that all look different but are grasped with a precision grip. And here are objects that look very different but are all grasped with a power grip coming from above. And then there are some objects like these horizontal cylinders that can be grasped by the monkey either way, from above or from below, as the monkey likes it. And that can then be combined in an experiment where we have a turntable where these objects are presented, and then a delayed grasping task where in the dark a fixation happens, a fixation point appears, then the object is shown for a brief time, then the light goes on, off, and then he has to wait for a variable time, and then the monkey makes a movement. And that is then shown here in a short movie where you see the time by that. This is happening here, and the multiple objects that are presented in the dark, and the monkey executes these movements. So to measure then the kinematics, we designed a magnetic-based data glove that has 5 degrees of freedom sensors on the fingernails, and 2 of them on the dorsum of the hand and 1 at the forearm, that allows us to measure all the degrees of freedom of the hand. So each finger has, as you know, 3 joints. The distal ones are 1-dimensional, and the base one is 2-dimensional. It gives you already 4 degrees of freedom. Same is true for the thumb. Gives you 20 degrees of freedom of the hand shape. And the location of the hand in space is 3 dimensions in direction and 3 dimensions in orientation. And if you take everything together, you have 1 dimension left, which is the elevation of the elbow, if you think about keeping the shoulder fixed. That is true, of course, for each arm, and you do kind of these manipulations with both arms seamlessly all the time. But we wanted to know what is exactly the hand shape of these grasping movements, and to relate that to then the neural activity. So here we see then the recording, tracking of the hand as the monkey is grasping various objects. And here you see the kinematics over time that can then be evaluated offline. If we do that, we can then classify the kinematics of the movement and see how many different grip types have been applied, for example, to these 50 objects. And here we see a classification that we have about 20 different classes of grip types for these 50 objects that I just showed you. So now we have an understanding of what kind of kinematics, or what kind of hand movements are happening in such a task. And we can then look at neural activity in these brain areas. I'm showing you microelectrode arrays, floating microelectrode arrays with 32 channels each, implanted in AIP, in premotor cortex F5 and motor cortex. So it gives us close to 200 electrodes, 200 channels, from which we can record and do spike sorting and then look at the signals. And if we do that, here is a summary plot where we see the percentage or the number of tuned cells on the y-axis, and the x-axis is time. The different colors are the 3 different brain areas. And this is for a subset of the objects where we see a variable kind of very different objects that are grasped in a different way and that look, of course, also differently. You see here in motor cortex, we have activity coming up strongly during the movement phase, when the grasp happens, and the hold phase. But in premotor and parietal cortex, we have already during the presentation, the visual presentation of the object, activity coming up and being persistent. But for example, here, we don't know, is this now a representation of the object, or is this a preparation of the action? And that's what we like to distinguish. And for that, we then look at the population. So for example, if we look at the area AIP in the parietal cortex, we can then take the number of neurons recorded from that area and then do canonical discriminant analysis and get then basically a PCA, so to speak, where we have then the representation of classification of these objects. So here we see the first and second component of the neural space as it clusters in the space. And here we have the kinematic space in comparison. And what you see right now is that it doesn't really fit. But still, if we look at these classes, and we look in 3 dimensions, we have clear clusters. And if we look at it carefully, we see that these clusters actually correspond to the objects. So we see here are clusters of objects and objects of similar size and different size of the same shape are clustered together. All right, that's here. And that is even persistent in the phase of the movement when the movement happens, so in the grasp phase. This is during the cue, when the visual stimulus is shown, and this is later in the grasp when the visual stimulus is long gone, but we still have the same clustering in AIP. So that can also be quantified with hierarchical clustering, gives us the classifications. So that means in AIP we have a clear clustering, at least in the first predominant components, according to the object shape. And you can also do a short control by using this set of objects that look very different, but are all grasped with a power grip from above. And we see here across these 6 objects, we have then a nice tuning percentage over AIP in F5 and nothing in M1 because the grip is always the same. So that's what I just showed you here in AIP. And if we look at area F5, we have, doing the same analysis, we would then look at trials that are recorded from parietal cortex, from premotor cortex, and look at the kinematics. We have then the kinematic space appearing here and the neuronal space here, do the comparison between the two. And what we see here is that there's a nice match, and there's this nice development of the clusters already during the cue phase and expanding further during the planning phase and the grasping phase and the holding phase. So we have a clear matching of the clustering in F5 with the kinematic space, and similar objects requiring different grips are located separately, and different objects acquiring the same grips are located closely together. So, and importantly, in premotor cortex, we have these motor features appearing already during the planning phase. And finally, if we do M1 analysis, similarly, we have then a match between the joint angle space of the kinematic data and the neuronal activity in the neuronal data. As you can see here in quantification. So together, we have then a representation with this neural activity in the 3 areas of these 50 objects according to the object shape in premotor cortex predominantly, and according to the grip type in premotor cortex and in motor cortex. I'm sorry, this was that. Okay. This is just what I said. So that doesn't mean, of course, that we have a high-dimensional population of neurons that no movement activity is represented in AIP, but what I'm saying is that there's a predominant representation of object shape in this area, and vice versa. So if we now want to ask whether we can decode these movements, there has been, so the idea would be that we have an understanding of how these neurons encode intended movements in parietal, premotor cortex, and motor cortex, and we would then interpret them using a decoder to control either a robotic hand or a virtual environment, and so on. We can ask how good this can be done on the basis of the recorded data as a first instance, and later on, of course, also in real-time experiments. This has been done for 20 years now, quite a bit. I'm showing you a video here from the lab of Andy Schwartz, who about 2008 had this paper in Nature where he trained a monkey to reach out in space using population vector methods to fetch marshmallows placed in different locations in space, and this monkey was able to move these marshmallows towards his mouth and eat them. So that was quite amazing that this is possible. But you can see that the grasp feature is quite rudimentary. The main aspect was on the reaching part of moving this robot hand in space, and that was quite impressive how well that worked. Then there was also human work by several people. This is a paper from the Donahue lab and Hochberg, where here a robot arm that was developed by the DARPA program is controlled by brain activity from a patient, and you see this patient is able to move this robot arm in space and can move light objects, even this little wooden cube. But you also see that the grip that is applied is kind of always the same, and is quite. It's not something you can take home to have a device that you can work with as a prosthesis at home, right? That's clearly experimental. Now, what can we do in terms of grasping using the data that I just showed you? If we have a simultaneous interpretation of the activity from one cortical array, as I just lay out here, the activity of the 32 channels, you can see when the monkey makes a precision grip and when the monkey makes a power grip, that the activity pattern in the population is quite different for these two grip types, and of course that's true for all the 20 grip types I just showed you before. And then the question is, how can we interpret that to make predictions? One simple one is a classification of the objects or the grip types from the data, from the population data. And if we do that here for these 50 objects, the question would be, can we predict the 50 objects from the neural population activity on a trial-by-trial basis? Here would be the real condition, one of 50, and here's the decoded condition. And the correct prediction would line up on a diagonal. And errors, of course, would be off the diagonal. And you see here, accuracy overall is about 50% correct. Chance level would be 2%. And you see the errors happen just off the diagonal. So there is confusion about different sizes of these objects. If this analysis is done a little bit later, it's slightly better, but it doesn't really change that much later in the task, I mean. But we don't want to predict objects, we want to predict the grip types. And so if we do the decoding of these 20 grip types, then things look much better. So we have, of course, less conditions. We have only 20 different grip types according to our analysis. Same confusion matrix. And here we see that we are about 86% correct in the performance during the preparation phase, and in the holding phase of the movement, it's above 90. We, of course, can then look at which brain areas code what, and this is then, of course, clear. If we take all neural activity together, we have a best prediction. But in the planning phase, premotor cortex is best. Motor cortex is lousy because it's not active yet. Whereas when it comes to movement execution, of course, all signals together are giving the best prediction, but then motor cortex is, as a single area, the best predictor of that. And AIP is not doing completely badly. We said before it's encoding objects, not grip types. But it's the worst area for decoding, but it's still not bad. And that means that even though this brain area is predominantly representing objects, it still knows information about what grip type to be made. And so that means there is, of course, cross talk between these areas, and in the subdominant projections, in subdominant dimensions, we see the representation, and that can be decoded. Besides the decoding of shapes, of hand shapes and objects, we can of course ask the question, can we also predict the hand kinematics continuously, as you might want to do it, that you move an arm and you kind of continuously control all the dimensions, that 27 degrees of freedom, and that's what we did in this study, where we here 1 degree of freedom is shown here, and that is predicted from M1, and it's a little bit less good predicted from signals from F5 and worse from AIP. That can be quantified, but I think a better way to look at it is when we just look at this video, where we see that if we look at the decoded kinematics in yellow, and in gray, the true kinematics that have been recorded while the monkey was doing the movement, you see quite a good match. So in this offline analysis, we can relatively well predict these grasping movements and the hand shapes in a continuous fashion as well. But nevertheless, and we have now ongoing work where we try to make this happening in real time. So the monkey has to be trained to make the real movements and grasp with real objects or in a virtual environment. But there is clearly some challenges that have to be overcome. The training of real-time decoders. So how do we start training the monkey to do these movements? And similarly, if we would go to humans, how do we train a decoder to represent these movements, given that you don't know the properties of the neurons in the population beforehand, in the human case. And one important thing is also the longevity of implanted electrodes that are good enough for doing electrophysiological studies in monkeys, because we have a lifetime between 2 and 5 years in our hands. But this is not something very appropriate for a patient, right? And if this device should be implanted in a patient, you would like to have a lifetime of such a device for 10 years or so, or even longer. And the reason for that is this is a foreign body, these electrode arrays. It's a foreign body, and they are encapsulated by a sheath of gliosis, and that pushes away or destroys the neural activity over time. The other thing which I don't mention today is sensory feedback. So there's this famous experiment by Roland Johansson who numbed the fingers of a subject with lidocaine, and then you can perfectly move your fingers because the muscles are in the forearm, but the sensory signals coming from the fingertips going to your spinal cord and brain are disrupted. And it's like when you go to the dentist and get a tooth repaired, you have a numb feeling, and that means you can still move everything, but you can't really feel what you're doing. The sensitivity of your hand is lost, and that makes it really difficult to manipulate in a delicate way objects. And so that is really important. If you think about using the hand for everyday control, we need to make it possible that sensory signals are also brought back to the brain or spinal cord. Okay, this brings me to the third part of my presentation, and which is a look at neural network activity. So as a first thing, we looked at a way to model the population activity that we have been recording in these 3 areas, where we have AIP and premotor cortex and motor cortex, and we wanted to basically have a model that basically takes as an input an image of an object and produces as an output the kinematics of the hand. And this can be done by a muscular skeleton model that we also trained or modeled on the basis of the kinematics, where we looked at the length of the 50 muscles that move the hand, an arm, and the output of this recurrent neural network model for grasping is then the control of this length of these 50 muscles over time. And the input is then an image, but we didn't want to have just a line coding by, say, image number 1 to image number 50. We wanted to be able to that the model is able to generalize, and that's why we implied a deep neural network. We started with ResNet and lately also used AlexNet for that, and used high-level features of this spatial convolution of these images, and use that as input to parietal cortex, to the modeled parietal cortex, the input module. And those are recurrent connections that are sparsely connected to the next module, and it's going back and forth. It's not so surprising that with a lot of tweaking, you are able to produce these movements, because after all, it's a neural network. We were able to do that with an overall error of about 5%. So you can train these networks that they see these images, identify the right movement, basically, and then do that movement over time. The only thing else they need is a starting or holding signal to make the movement start at the right moment. But to make this then biologically similar, we had regularization properties which put the cost on firing rate, and cost on input-output connections, and the cost on complex state trajectories. And with that, we were able, and that was something which we net composed, we saw then that the neural data and the module neurons looked in their population quite similar. So we did this with canonical discriminant analysis, where we see canonical variables, where the first 7, they explained about 80% of all the variability, and the first, you see, the correspondences are quite high. So that means this model is capturing the features of the real neural network quite well. And the other thing what we did was to look at functional connectivity between identified neurons in this population. This work that Benjamin Dunn did in my lab. So we have this 6 arrays of electrode arrays with about 200 electrodes and about the same number of neurons isolated. And he did really a painstaking job in making sure that he's recording or identifies individual neurons, not clusters of multi-unit activity. And then looked at the cross-correlation between these identified neurons to generate a matrix of connectivity by looking at the cross-correlation. And that's what you see here. And with that, you can do modules, generate modules of connectivity like this one and the small one in between. And you can also then look at the topography of this set of neurons, how they are connected, together with collaboration of Olaf Sporns. We used that toolbox and then showed at the connectivity across the population. What we found, of course, is the chance of observing a functional connectivity between with one pair of neuron is highest when these neurons are close together on the same electrode. The further they are apart, the less the likelihood. Here on the same array, on the same area, and then across different arrays. But nevertheless, if you look at then these modules, how they fit together, you see that not all neurons belong from one cluster, from one module, belong to the same area. They have long-range connections as well. And then this way. I'm sure. And the interesting thing what we found was that there are neurons that are much more strongly connected to other neurons than the majority, and those are so-called hub neurons. They have strong connections to all other neurons, and those hub neurons are important for the communication, right? And they form also a rich club in the sense that they are more strongly connected to each other. So hub neurons connect to hub neurons much more often than they should be on a stochastical way. And the other thing which was quite interesting was that looking at the oscillations in the connections between these neurons in the cross-correlation frequency bands, we found that these hub neurons are very often predominantly oscillating either in the beta frequency range or in the low frequency range, whereas local neurons did not have that oscillatory property. So that particularly the hub neurons are oscillatory active, and that might go in line with the other theories of oscillatory activity along cortex. And I think I stop here. Thank you very much for your attention. And this is the work that was done by Benjamin Dunn, the analysis of connectivity, by Jonathan Michaels, the recurrent neural network analysis, and Stefan Schaffelhofer, the work on the population activity in the grasping. Thank you very much.
There is time for discussions, the two, before, and then you.
I had a question about your recurrent neural network that you trained to do the grasping. I mean, after you extracting the feature with the, I think, ResNet, and then you pump it to the recurrent neural network, I was wondering about the recurrent neural network. Did you inspire for designing that recurrent neural network from the actual neural substrate, or you start from a basic connectivity and just train it to do the job that you want?
Yes, the deep convolutional network is fixed, right? That's trained before, and we just use it as it is. It's basically a device that it identifies images, and we use the higher level features as the input. And then here, the RLN are randomly connected within each module, and then that is trained. So that is then, together with the regularization parameters, optimized.
If you allow me, two questions. First of all, beautiful work, Hans-Jörg. I should really say that very clearly. I assume in your experiments, objects are always presented in one and the same location. So in other words, the distance is constant. I was wondering if you find, or if you would expect to find information specific for reaching out to the object, or if you would say, well, in order to fully control the sequence of reaching out and grasping, you would have to place yet another electrode array, let's say in the parietal reach area. That's one question. And a second one, I think it was Bob Woods who emphasized yesterday that the brain offers a large degree of redundancy. With respect to reaching out and grasping, there's yet another obviously completely independent representation on the medial side of the hemisphere. So is it just redundancy, or would you, well, try to speculate that there are distinct functions that distinguish the two systems?
I mean, the experiment has already been done, right? The grasping areas have been inactivated. And then you see, like Leo Fogassi did that, and in parietal, and Murata in premotor cortex, or vice versa. Then you see a predominant deficit in grasping, but not of reaching. In these, if you inactivate AIP and F5, so that would already argue that these signals that we see here in these areas, only this one would be heavily, as a function, would be heavily being affected. Whereas we still see these other properties. So that could be that what we see here is a high-level efference copy that is maybe used to make computations in these areas as, you know, information input, but it's not used to make the functional output, at least as a first approximation. So that could be that there is not really every area in the brain doing everything, but every area doing something in the brain needs a lot of information for contextual information. I think that was the second question. What was the first one?
The first one, information on object distance.
Object distance. Yes, I think that would be just adding another feature, right? I try to show here that we tried very hard to come up with new ideas, what else can be represented in these brain areas for grasping. There have been experiments in other brain areas, like in PO, that we just change the distance of a reach, and then you would see a similar tuning for the distance of the target. I'm quite sure if we would do the experiment in these areas, we would see similar things.
But at least the clinical cases suggest that there is a differentiation between reaching out towards objects and manipulating them, right?
Yes.
If you, for instance, take optic ataxia,
But what I think is going on is that these areas that are highly important for planning and executing grasping movements, they have access to information on other features, like the reach position, the gaze position, the amount of force that needs to be applied, and maybe other things that we haven't even thought about.
Yes. Last question.
Firstly, thanks for the very nice talk. And I have a question about this famous study which is done on rodents. Through lesion studies, they have concluded that the motor cortex is necessary to learn new motor activities, but it is not necessary to perform motor activities unless they need dexterity. So I'm curious to know if you have studied the same thing on primates, or if you have comments, because the implications from the study is that for simple motor tasks, possibly there are some alternative subcortical paths which can provide that sort of neural pathway. So motor cortex is not necessary for that.
I think it depends a lot on what kind of task you talk about. For primates, the dexterity of finger movements is strongly associated with the cortical motor neuronal projections that have been shown in primates strongly, and that goes predominantly from motor cortex to the straight to the spinal cord in the cortical spinal tract, of course, as has been studied by people in the past anatomically. And there are a few projections from premotor cortex too, so it's not completely exclusively motor cortex, but it's about only 5%. Of course, there are other indirect projections, as you mentioned, which is good enough for more simple movements like trunk movements or reaching movements. So if you have a lesion in exclusive lesion in cortical motor neuronal pathway, you still be able to make reaching movements with the arm, but the fine finger dexterity might be lost. And that's also what you see in stroke patients, that the dexterity of the finger movements is lost first in these problems. As far as to learning of new skills, there's probably a lot of adaptation going on when you adapt to stroke rehabilitation or so. So I think there's still a lot of learning going on when you have a compromised cortical spinal tract. But so I would probably think you still can learn new things, but you are starting with a deficit.
You are very lucky because you got more time, because we should stay a little bit more inside here because of maybe something going on outside. So we have some more 5 minutes to talk.
Right.
So who has more questions, or probably also to the also a chance to the first speaker, if you want. Okay.
Thanks for this very nice presentation. You showed that the functional connectivity is particularly effective on certain units between the nodes. Now my question would be, are the type and strength of functional interactions helpful for the decoding? Is there some relation between what the networks really do and not only the units in their activity?
Yes, that's what we are after right now. The study that I showed you here basically ignored the entire fine structure of the task. So we kind of took all spikes of the entire session together, or at least to the entire task concatenated together of the entire trials, not looking at particular epochs of the task, not looking at particular conditions of the task. This was necessary because we needed a lot of spikes to do the spike-spike correlations. But we're now looking at spike field coherence, and where we actually exactly doing that. So this is Swati Sesatri, who is doing a pairwise phase consistency analysis of this similar data. And what we see here, that there are changes in time. So we see here for different tasks, we see here different features, spike activity and LFP activity, and the correlation across them. And then we can look at the different properties at different areas and how they change over time during the task. So that's the next study, what we want to do. And then the hope is that we can identify the hub neurons as the one that are maybe important for the backbone, and then other neurons that contain the information for the transportation. So the hypothesis that we have right now is that there is a difference between the structure of information communication and the information content. But we're not that far yet.
Now we have the nice situation that we should go on with the next talk.